Hi, my name's Kira, and Gabby and Sydney and I are part of CBE Team 7, and our project is entitled Food Production Without Photosynthesis. For a little bit of background and context on our project, the global population is currently 8.1 billion people and is estimated to surpass 9 billion by the year 2050. Additionally, 25% of all land on Earth is used for permanent meadows and pastures for raising livestock for animal protein or byproducts. Additionally, farmed food production, such as soy crops, are highly dependent on water availability, arable land usage, sunlight, and climate conditions. So, in light of both the rapidly growing global population as well as a changing climate, it's imperative that there's enough protein to sustain the world's growing population. Therefore, single cell protein or SCP production could prove to be an attractive avenue to supplement protein in the diets of people all around the world. SCP is defined as protein-rich biomass that's derived from unicellular microorganisms such as bacteria, yeast, or algae. SCP is an attractive alternative to farmed protein sources such as animal meat or soy because it occurs within a closed system and requires significantly less arable land and water than these two different types of protein sources. And in our specific project, we designed an SCP production process that utilizes the type 1 obligate methanotrophic bacterium called Methylococcus capsulatus. In our project, we designed and evaluated the feasibility and societal impact of a manufacturing process to produce bacterial SCP as a human food grade product. The plant can produce 62,000 tons of SCP per year, which would make it the largest continuously operating SCP production plant in the entire world. Additionally, it would be the only bacterial SCP production process that would make human food grade product. So, because this is a very new process, it was important to scrupulously design both the equipment and the process in order to ensure that the employees and the people consuming the final protein product would be safe. In order to operate the process safely and effectively, it would need to be sterile in accordance with FDA guidelines. And additionally, because this process would produce human food grade protein rather than protein for animal feed, it was important to add additional unit operations to purify the final product to make it human food grade.
Methanotrophic bacteria can use either methane or methanol as carbon sources, which they metabolize into protein-rich biomass. We designed two processes, one with a methane feed and one with a methanol feed, in order to evaluate the feasibility and profitability of producing 50,000 US tons of SCP a year. With the average American consuming a tenth of a ton of protein a year, this amount of protein can contribute to the yearly diets of hundreds of thousands of Americans. To ensure that the SCP doesn't trigger any allergic reactions upon consumption, the cells will be mechanically lysed, destroying the immunogenic properties of the intact cell wall. Additionally, the nucleic acid content of the biomass, in essence, any DNA or RNA that's been left over from the cells, must be reduced. In particular, the purine content must be reduced to within a healthy and acceptable limit, since purines can contribute to adverse health effects such as increasing the risk of gout. The purine content of our final product will be lower than other purine-containing foods such as red meat. Purifying the protein biomass of excess harmful nucleic acids is most effective in membrane ultrafiltration, a separation process where suspended protein.
Agriculture alone accounts for approximately 50% of global land usage, 30% of greenhouse gas emissions, and 70% of freshwater usage. Plus, it takes almost 100 times as much land and 10 times as much water to produce 1 gram of an animal protein like beef compared to a plant-based protein like soy. 60% of global protein intake comes from plant-based sources, while the other 40% comes from animal products. Over a third of that is consumed by the wealthiest fifth of the globe. Further, over half of the protein consumption in the United States comes from animal products, which equals 3 times the global meat consumption per capita. These numbers will only continue to grow. The urbanization and overall economic growth across the globe correlates with an increase in consumption of animal products. Plus, as humans age, their protein requirements increase. With the overall global age increasing, protein needs are growing in tandem. Overall, these factors create a global protein requirement that is unsustainable and unmet under current conditions and will continue to worsen as the population continues to grow. Although an increase in plant-based proteins provide more cost-efficient and environmentally friendly solutions to meat production as it relates to the global food crisis. They often lack the complete range of essential nutrients necessary in a human diet. Specifically, they lack some essential amino acids and nutrients such as zinc, iron, and vitamin B12, the lack of which have been known to stunt child growth. This means that plant proteins are considered lower quality protein sources than their animal counterparts. Additionally, although many plant-derived proteins are on the market, many of them are legume derivatives coming from peanuts, soy, and peas, which are all common allergens. Thus, it's imperative to develop a novel protein source to support the increased need as well as withstand the effects of climate change. SCP products provide a solution that's free of common allergens and is comparable to high-quality proteins as they're naturally rich in digestible indispensable amino acids, or DIAAs. Plus, SCP uses significantly less arable land and fresh water than traditional agriculture. For example, Callista's feed kind uses approximately 100 times less fresh water and 1,000 times less arable land compared to soy on a per ton basis. Since SCP is grown via fermentation in a closed environment, it's also independent of many unpredictable risks associated with traditional agriculture, such as pests, pathogens, environmental and soil degradation, extreme weather events, and climate change. Therefore, because the methanol-fed process is economically and technically viable, it should be considered in order to alleviate some of these issues related to the need for increased and sustainable protein production.
We found that the methanol-fed process would be economically feasible, with the final plant design having an annual production capacity of 62,000 US tons. The proposed location for the plant is Groves, Texas, and the product's selling price is $7 per kilogram, based on the average $10 per kilogram price of 90% soy protein isolate. This 30% reduction in price will position our protein product competitively against soy, which is a much more familiar protein source. The internal rate of return for this process was found to be 29%, with a return on investment of 28%. The net present value is $90 million. Thus, we recommend this plant be considered for development.